Hello everybody and welcome back to our last session on the genetic code translation and protein biosynthesis. I have already shown you this slide in a slightly different context, but this slide actually summarizes quite well what we need to translate the message of the mRNA into proteins. First of all, we need mRNA. In addition, we do require ribosomes and tRNAs. So let us have a look at the role and impact of tRNAs on the translation. tRNAs function at specific site in the ribosome during their translation. And what you can see here is a schematic view of a tRNA molecule. Again, similar to other nucleic acids, tRNA are polar. So we have a 3' end and also a 5' end. In addition to those two ends, another important feature of a tRNA molecule is the anticodon. And the anticodon binds to the codons on the mRNA in a complementary way. There are also a few additional features on the tRNA molecule that are actually not that important for you, but just to give you the full picture, tRNAs also contain the so-called T-loop and D-loop, which are important from biochemical point of view, but this is not something you should be worried about at this stage. What you need to remember though is that because the genetic code is degenerated, less than 61 tRNAs are needed for the 61 codons. In bacteria, this is slightly less, but in this module, we are mainly focused on the human body, so 61 codons is the number you need to keep in mind. So what you can see here is a cell. And I hope that you remember the role of those individual organelles in the cell from our cell biology lecture. The site of protein biosynthesis and translation in those cells are ribosomes. And ribosomes can be either present in the cytoplasm as free ribosomes, or they can be attached to the endoplasmic reticulum, more specifically to the rough endoplasmic reticulum. And those organelles are in charge of translation. So how do ribosomes look like? Ribosomes are composed of two major parts. The small ribosomal subunit that you can see here and the large ribosomal subunit. So what are the different jobs those subunits do during translation? The small ribosomal subunit reads the RNA. And we also do have the large subunit, and the job of this subunit is joining amino acids together to form a growing polypeptide chain. Each of those subunits consists of ribosomal RNA and also ribosomal protein. So, are there any differences between bacterial ribosomes and eukaryotic ribosomes? Indeed, they are. And the first obvious difference is that those ribosomes have different size. In eukaryotes, those individual subunits tend to be slightly larger. In the eukaryotic ribosomes, the large subunit is 60s large. In contrast, in bacteria, this is 50s. There are also differences regarding the small subunit, which is 40s in eukaryotes and only 30 in prokaryotes, that means in bacteria. And this is actually quite important. They not only differ in size, but they also differ in structure. This is especially important if we think about ribosomes as targets for antibiotics. And indeed, antibiotics are very, very important. The World Health Organization has recently warned of the global crisis over antibiotics resistance because many bacteria are developing those. And I know that you're going to be hearing a lot about antibiotics and bacteria in Dr. Black's lectures. But for this lecture, please keep in mind that ribosomes are quite frequently targets of antibiotics. An example of such antibiotics targeting bacterial ribosomes are aminoglycosides, including streptomycin, kanamycin, neomycin, and so on, and also tetracyclines. They target the small subunit of bacterial ribosomes. In addition, there are also antibiotics targeting the large subunit, including erythromycin and azithromycin. And finally, there are antibiotics that prevent proper interaction of those two subunits, thereby stopping translation in bacteria. And those antibiotics are, for example, oxazolinoids, like including Zyvox, which is the brand name of linezolid. And again, you will hear more on antibiotics in Dr. Black's lecture. So let us have a look at ribosomal subunits in translation a bit closer. In order to understand translation, you need to understand at least the three different structural features of ribosomes, namely the amino acyl site, peptidyl site, and exit site, short E, P, and A. And those are structural features on the large subunit of the ribosomes. And again, this subunit of the ribosomes is in charge of assembling amino acids into a growing polypeptide chain, whereas the small subunit reads the mRNA codons. And this is how it looks like. We have the mRNA, which is bound to the small ribosomal subunit, and after assembly of the complex, the large ribosomal subunit is in charge of producing the growing polypeptide chain. So how does translation look like step by step? The first step of translation is initiation. What happens here is that the first tRNA recognizes a start codon and binds to the respective codon on the mRNA. The mRNA is already bound to the small subunit. Once this happens, the large subunit can enter the complex, and now we have a full and complete ribosome. So as you can see here, we can see the mRNA binding site here. We have the P site and the A site as well. So those are the sites on ribosomes we need to pay attention to. During the elongation, once the translation has been initiated, the tRNA, which is bound to the first codon, actually moves to the site, right? Opening thereby the third codon. And then the first tRNA in the complex is getting released at the exit site, right, which is here. Afterwards, the next tRNA carrying an amino acid can enter the complex. The last step in translation is termination. And termination is induced by the so-called release factor, because release factor is able to bind to the stop codon on the mRNA. So what's here quite interesting is that although those stop codons are triplets, just like very ordinary codons, they are not decoded by tRNAs, because the release factor is a protein, and this is quite unique. Once release factor is bound to the mRNA, this leads to disassembly of the complex, and we have again a free large ribosomal subunit, a small ribosomal subunit. We have the mRNA, which is now free, and we have the released tRNAs, and finally our peptide, which is now ready to be used. So what happens after translation is complete? Obviously, this is not the end of the story, because proteins are quite different and they can be modified. For most proteins, translation is only the first step in the life cycle. because moderate to extensive post-translational modifications are required before a protein is complete, and this happens at the Golgi apparatus. Some polypeptide chains require the addition of other molecules. Other must be spliced or digested during proteolysis to be fully functional. And often this involves the excision of the first amino acid in the chain, which is usually methionine, as this is the particular amino acid indicated by the start codon. In reality, this is quite complex, and you don't need to remember this for the exam. This is just to give you the whole picture. After the proteins are produced, following on transcription, processing, and translation, they can undergo quite extensive post-translational modifications. And indeed, there are over 200 possible modifications on proteins. Proteins can be phosphorylated, they can be glycosylated, that means that there are sugars added on, they can be modified by attachment of lipids, and they can also be cleaved. They can be made shorter by process called proteolysis. And all of those modifications do have effects on catalytic activity of proteins, on the ability of those proteins to associate with other proteins, on the stability, half-life, localization, and so on. So this step is also quite important. And you will learn more about post-translation and modification of proteins and about proteins in general in a follow-up lecture, which is going to be delivered by Dr. Cottrell. However, before you will have the opportunity to look at this in detail, I would like to talk briefly, at least as a sort of introduction, about protein folding. Because proteins are synthesized on the ribosomes as a linear polypeptides, but they do require proper folding in order to function properly. And again, you will hear more about this in Dr. Cottrell's lecture, but all you need to remember here is that there are different levels of structure of proteins. We have the primary structure, which is dictated by the order of amino acids in the polypeptide. We have the secondary structure, tertiary structure, and quaternary structure. And the activity of proteins depends on the integrity of the final tertiary structure. Folding is also important. I'm going to be going through this fairly quickly, because folded protein have lower level of energy. In contrast, unfolded proteins have very high energy and high level of entropy. They are therefore unstable. So in order to make those proteins stable, proper folding is needed. And the molecules in charge of folding of proteins are called chaperones. They are a class of proteins that aid in the correct folding in vivo. This is how those guys look like. They look sort of like a barrel. Unfolded proteins are able to enter this complex. Then chaperones require energy in the form of ATP, and they assist in protein folding, resulting in fully functional and folded polypeptide. As we have heard now, folding is really important to produce proteins that function properly. But what happens if a protein is unfolded? This basically produces not a useful protein for the cell, but a toxic protein clump. Diseases can be often caused by such misfolding of proteins. This actually highlights why this is also an important step in protein biosynthesis. And examples of those disorders are Kuru disease, Creutzfeldt-Jakob disease in humans, Scrapie disease in sheep, or mad-cow disease. So what I would like to do now is to show you the symptoms of Scrapie in the sheep. As you can see here, the animal is moving in a slightly uncontrolled manner, is not able to control the movement properly, and this is all due to protein misfolding. Because those toxic clumps of proteins are affecting the brain. Similarly, in human, protein misfolding within the brain can cause neurodegenerative disorders, including Kuru disease. So what you can see here on the left-hand side is a brain of the patient suffering from Kuru. If you compare the size of the brain to a healthy control brain, then you can see there is really high level of neurodegeneration going on. And similar to the symptoms of scrapie, patients with this disease lose control of motor function in their body. They lose cognitive abilities, and this is due to neuronal degeneration caused by faulty protein folding. If you look at the cerebral cortex from a patient affected by a neurodegenerative disorder resulting from prions or protein misfolding, then you will see that those brains look quite spongy with a lot of neuronal cell death going on. And the simple causative cause here is protein misfolding. And indeed, there are many disorders in humans resulting from protein misfolding, and this is not limited to Kuru or Creutzfeldt-Jakob disease. And there are many examples of those disorders in the web. With this, I would like to end up our lecture, and I will see you in our next genetics session fairly soon. Thank you very much for your attention.